Hello friends, welcome back to my YouTube channel Nesting Padasalai. In this video, we are going to discuss top 25 expected questions regarding to BARC OCES examination. So first question, definitely one question will be coming from ion exchange chromatography. A lab is trying to purify a histone binding protein from its native source. Which one of the following purification techniques should be used for one-step purification of a protein? Okay, so histone is a basic protein. So histone is made up of positively charged amino acid which include lysine and arginine. Okay, so histone is involving in packaging of your DNA. Okay, so histone is a positively charged protein. Okay, histone is a basic protein. Okay, so then which chromatographic technique you will be employing? The correct answer is cationic exchanger chromatography, which include cationic exchanger. So these cationic exchanger, they will be having a negatively charged group with them. So these negatively charged group will attract the positively charged cation. So histone is a positively charged cation. So you will be purifying histone by cationic exchange chromatography. Okay, so for positively charged protein and basic protein, you need to employ cationic exchange chromatography. Okay, whereas this anionic exchanger will have a positively charged group. They will be attracting the negatively charged anion. Okay, the next question is from bioinformatics. So here the question is that number of unrooted trees in a phylogeny of 5 sequences. Okay, so this is a formula of rooted trees. So 2n minus 3 the whole factorial divided by 2 to the power of n minus 2 multiplied by n minus 2 factorial. Okay, so here the question is that number of unrooted trees. Okay, so what is the formula to find number of unrooted trees? It is 2n minus 5 the whole factorial divided by 2 to the power of n minus 3 multiplied by n minus 3 factorial. Here n is 5 sequences. Okay, so you need to apply and you need to find. For this question the correct answer is 15. So here you can able to, I had solved you the question. So the denominator, it is for 5 minus 3 factorial alone. Don't take it as a whole factorial. First you need to solve for 2 to the power of 2, that is 4, and here 5 minus 3 is 2 factorial. So 4 multiplied by 2, which is 8. Okay, so that's how you need to solve this question. Okay, so next question is every neuron in the body develops from. Okay, so you need to know about 3 germ layer. From these germ layer, different parts of the body will be coming. Okay, so first is ectoderm. All nervous tissue will be coming from ectoderm. So ectoderm is classified into neural ectoderm. Okay, so from neural ectoderm, your neural crest cells will be coming. So here the question is that from which part, from which particular thing neurons will be originating. So neurons will be coming from neural crest cell. Okay, so neural crest cell is a derivative of neural ectoderm. So these are the derivative of neural crest cell. So from neural crest cells, peripheral nervous system will be originated, endocrine paracrine derivatives will be coming, your pigment cells will be coming, facial cartilage and bones will be coming, connective tissues will be coming. Okay, so from the peripheral tissue, that is from peripheral nervous system, all your neurons will be derived from this particular peripheral nervous system. So correct answer for this question is neural crystals. Okay, so ectoderm, your nervous tissue, epidermis will be coming, cornea and lens of eye will be coming, enamel of tooth will be coming, and melanocytes will be coming from this ectoderm. Whereas from mesoderm, your skeletal, smooth cardiac muscles will be coming, blood, bone marrow, lymphoid tissues will be coming, and endothelium of blood vessels will be coming, and other organs of urogenital system like your ureter, kidney, gonads, reproductive duct, everything will be coming from the mesoderm. Okay, whereas from endoderm, your epithelium of digestive tract. So you should know epithelium of skin, epidermis, and epithelium of oral and nasal cavity will be coming from ectoderm, whereas epithelium of digestive tracts will be coming from endoderm. Okay, so the next thyroid, parathyroid, thymus gland will be coming from this endoderm only. Okay, so the next question is 5' end of mature transfer RNA has. Okay, so if you study about the post-translation modification of this transfer RNA, you can able to know 5' end of mature transfer RNA will be holding a single phosphate group. So 5' end of mature transfer RNA will be having a monophosphate group. Okay, whereas 3' group will be having a OH group. In this 3' group, you can able to find a unique sequence called CCA. Okay. The next is with regarding to BLAST program. Which one of the following BLAST search program is used for identifying homologs of genomic DNA query? So query is your DNA in a protein sequence database. Okay. So BLAST-N is a program which you can use when the query type is nucleotide and the database. Okay. So you are having a nucleotide sequences and you are going to search your nucleotide sequences in nucleotide database, means you can able to use this BLAST. Whereas this protein, BLAST-P is for protein. You're having an amino acid sequence and you're going to search your amino acid sequence in a protein data bank, means then you can use this BLAST-P. Okay. Whereas T-BLAST-N means you're having a amino acid sequences and you're going to search that particular amino acid sequences in a nucleotide database. Okay. So in that case, you need to employ this T-BLAST-N. Okay. Whereas blast x means you're having a nucleotide sequence data, that is you are having your query is DNA and you're going to search your nucleotide sequence in a protein data bank, means protein database, means you need to employ this blast x. Here the query type is genomic DNA and you're going to search in a protein data bank, which means you're going to employ BLAST-X program. Okay, BLAST-X is for searching nucleotide query sequence in a protein data bank. Okay, which are the following modes of inheritance seen in cystic fibrosis? Okay, so the inheritance pattern of cystic fibrosis is autosomal recessive. Okay, so autosomal dominant include Huntington disease, neurofibromatosis, Marfan syndrome and other disease will be following a autosomal dominant. Whereas the Tay-Sachs disease, sickle cell anemia, cystic fibrosis, phenylketonuria, all these diseases will be following a autosomal recessive pattern. Okay, so you need to study about the example of disease also. Next is which one of the following is the best suited for hydrolyzing peptide bond on a carboxyl side chain of aromatic amino acids. So aromatic amino acid include phenylalanine, tryptophan, tyrosine. So here you can able to see the trypsin, which is from bovine pancreas, will be cleaving at lysine and arginine amino acid at the C-terminal end. Okay, which means they will be cutting in the right hand side of the lysine and arginine amino acid. Okay, whereas the submaxillaris proteases, which is isolated from mouse submaxillary gland, it will be cleaving at the C-terminal side chain of arginine. Okay, next is chymotrypsin, which is also from bovine pancreas, will be cleaving at C-terminal side chain of your aromatic amino acid. Okay, next is Staphylococcus aureus B8 protease. It will be cleaving at the C-terminal side chain of your acidic amino acid. Okay, whereas this asparagine N protease, which is isolated from Pseudomonas flaggi, will be cutting at N-terminal side. Okay, the Staphylococcus aureus B8 protease will be cutting at C-terminal side of acidic amino acid, whereas this asparagine N protease will be cleaving at the N-terminal side of your asparagine and glutamic acid. Okay, next is pepsin, which is isolated from pig stomach. It will be cleaving at the N-terminal side chain of your aromatic amino acid. At the same time, it also have a capability to cleave at N-terminal side of your leucine. Okay, so next important thing is cyanogen bromide, which is actually a chemical reagent. It will be cleaving at the side chain, carboxyl terminal side chain of methionine. Okay, so here they are asking about the carboxyl side chain with which particular thing is involved in cleaving in carboxyl side chain of aromatic amino acid. The correct answer is chymotrypsin. Next is which substitution matrices should you prefer in order to find a distantly related orthologs through BLAST search. Okay, so here you should know about BLAST search. Okay, BLOSUM matrices with high number and PAM matrices with low number are designed for comparing closely related sequence or more conserved sequence or less divergent sequence. Okay, for example, if you are going to compare a nucleotide sequence between rat and mouse, then you should use BLOSUM matrix with high number and PAM matrix with low number. Okay, next is for distantly related protein. So for distantly related protein, what kind of thing you will be using? Okay, BLOSUM matrices with low number and PAM matrices with high number will be generally employed for comparing distantly related protein or more divergent protein or less conserved protein. If you are going to compare an amino acid sequence between rat versus bacteria means, then you should employ BLOSUM with low number and PAM with high number. So here they are asking which substitution matrices you will be preferred in order to find the distantly related ortholog sequences. Okay, so distantly related means they are more divergent, at the same time they are less conserved. In that case, you need to employ blossom with low number and PAM with high number. Okay, so among this blossom, so correct answer is blossom 40 and PAM 250 is the correct answer. So blossom with low number and PAM with high number is generally employed for comparing the sequences with distantly related organism. Next is in Ramachandran plot, the value of dihedral angle psi. So you should also note down the symbol. This is a symbol for psi. Is based on rotation around. Okay. So here you should know phi angle is an angle of right-handed rotation around N and C alpha bond. Okay. Whereas the psi angle is an angle of rotation around C alpha and C bond. Okay. Psi CC. Okay. Just remember like that. And omega angle is an angle of right-handed rotation around CN bond. Okay. So CN, just remember cyanogen bromide. Okay. So here the psi angle is based on rotation around C alpha and C bond. Next is CDK2 cyclin E function in. Okay, so these are the list of CDKs, the cyclin partner, and in which phase they are generally employed. Okay, so in G1 phase, CDK4 and CDK6 along with the cyclin D will be engaged in regulating the activities in G1 phase. Okay, whereas in G1 to S phase, they will be in that particular phase, cyclin 2 and CDK2 and cyclin E will be taking place, they will be taking care of the regulatory aspects of this particular G1 to S phase. Okay, in S phase, that is in synthesis phase, the mechanism of regulation is provided by CDK2 and cyclin A. Whereas in G2 to M phase, the CDK1 along with cyclin A will be taking care. Okay, in mitosis phase, CDK1 and cyclin B will be taking place. Okay, so here I am asking CDK2 and cyclin E is functioning. Okay, so CDK2 and cyclin E is functioning G1 to S phase transition. Whereas CDK1 and cyclin A is responsible for G2 to M phase transition. Okay, so you should note about that. In G1 phase, CDK4, CDK6 along with cyclin D will be taking care. In G1 to S phase, CDK2, cyclin E will be taking care. In S phase, that is in synthesis phase, CDK2 and cyclin A will be taking care. In G2 to M phase transition, CDK1 and cyclin A will be taking place. In mitosis, CDK1 and cyclin B will be taking place. Okay, we will be discussing few other questions regarding to this important question series in next video. Thank you friends. Thanks for watching this video.